Hi everyone, this is Computational Cardiology, and today we're going to be going over systemic circulation. We've been talking a lot about the heart and the way that it works, the way that it interacts with the body, but let's actually talk about where we've been talking a lot about the heart and the way it interacts with the body, but we haven't really been branching beyond that. So let's take a look at blood and where blood goes. Let's talk about blood. Some of the main components of blood are red blood cells, and these transport oxygen from the lungs to the organs and then carbon dioxide from cells as waste products back towards the lungs so that they can be exhaled. Blood also contains a lot of nutrients, and these go essentially from the liver throughout the body. It contains white blood cells and antibodies, and these go to and from infection sites, as well as hormones, which go from the endocrine system to the rest of the body. There are a few main vessels or vasculature components for blood. So these are the arteries, and there are a few main vasculature components for blood to travel through, and each one has its own main purpose. According to the lecture slides, arteries have the main purpose of giving off fast flow with little pressure drop. Arterioles, which are essentially branches of arteries, have a large pressure drop, and this is to control flow. Capillaries, which branch off from arterioles, enable diffusion. And then from capillaries, they kind of branch into venules, and venules serve to provide capacitance. And then venules turn or branch off into veins, whose main job is also to provide low resistance. If we take a look at some ranking of these different vessels or vasculature components, the largest diameter is in the vena cava, and this is where blood enters the heart through it. The vena cava, SVC and IVC, superior and inferior vena cava, both branch into the right atrium. Then the next largest diameter is the aorta, and then it's also important to know that the smallest diameter is the capillaries. The highest cross-sectional area goes to the capillaries, and I was a little confused by this at first, but even though they're really small individually, there are so many capillaries in the body that as a result they end up having the highest cross-sectional area. And then it's also relevant for the class to know that the vena cava and aorta, in that order, have the smallest cross-sectional areas. In terms of average pressure, there's the most pressure in the aorta, then arteries, arterioles, capillaries, venules, and veins, and then vena cava way at the bottom, and that makes sense, right? Because the vena cava and the veins are getting blood not directly from the heart. This is after the capillaries and after there's been that large drop in pressure in the arterioles. And then the largest negative drop in pressure, so the largest drop in pressure, as we just mentioned, goes to the arterioles and small arteries. As far as volume, there is the most volume in venules and veins, and then with a question mark, I wrote down that capillaries have the second most volume, and again, we're following the logic that they're small, but there are a lot of them. And then finally, for velocity, the fastest velocity is in the aorta, and then in the vena cava. And then finally, all the way down, the slowest is in the capillaries. And that makes sense because capillaries exist so that diffusion can occur. You don't want blood to be speeding through there. You want it to go pretty slowly. Keep in mind, though, that even though there are these differences in volume and velocity, flow is consistent throughout the entire system. That's because it's a function of cross-sectional area and velocity. So where there's a low cross-sectional area, there's a high velocity, and where there's a high cross-sectional area, there's a low velocity. If we look at the structures of vessels, most vasculature is made up of an endothelium on the innermost component. So the way I drew this, it's kind of like this is the lumen of the vasculature, and then we start moving out as we go down. So the endothelial cells interact directly with the blood, and then elastic tissue goes around those, smooth muscle around the elastic tissue, and then finally fibrous tissue. This looks a little bit different in capillaries. They just have endothelial cells. And then in venules, they have endothelial cells and then fibrous tissue right below that. There are some mathematical relationships that are important to know. These get asked, at least in my experience, on the exams and on the homework. And to do this, you don't necessarily have to have Poiseuille's law memorized, but it's a good idea to kind of understand the relationships. So you might remember this from biological models and systems sophomore year. One of the relevant ones that we got asked about, at least, is related to the relationship between flow pressure, and radius. So look over here and you see that flow is related to the fourth power of radius. So if the radius decreases to 0.8 its own size, so by 0.2, the flow is actually changing by 0.8 to the fourth. So you end up having roughly 40% of your original flow. And then if you want to increase the pressure to cancel out for that decrease in flow, so the flow doesn't actually change, so you're canceling out for the decreased radius by increasing pressure, you have to increase it by a factor of 0.8, so the new radius to the fourth, and that comes out to around a 293 millimeter of mercury difference. There's a chart that Dr. Haas goes through in class, but what we're essentially getting at here is that even a very, very small change in radius can lead to this huge, huge change in pressure. I mean, if you take your blood pressure and it's 293 millimeters of mercury higher, you're in trouble. So this is relevant for diseases like atherosclerosis, where part of your vessel or part of your artery, for example, your vein can get blocked off. If there's even a relatively small change in radius, your blood pressure is going to shoot up, and it's important to think about what that does for the afterload of your heart, where you're over-exercising the muscle, so it's more likely to fail. And just for fun too, I included the 0.2 radius. That means that your flow, your new flow, is 0.0016. The amount of your original flow, and your pressure would have to jump by 625 millimeters of mercury to account for that change, which is insane. But moving on, let's look at controlling resistance. So your vessels and your vasculature have receptors in place. They have muscle that can contract and dilate, and this all essentially helps account for these changes in pressure, so that when you stand up, for example, you don't immediately pass out. So there's local control, and this is a function of metabolic and myogenic autoregulation, related to shear stress. You have carbon dioxide as a vasodilator in most of the cells. So, for example, carbon dioxide, for the most part, is a systemic vasodilator, where it encourages your vessels to open up a little bit. But this metabolic control can also be a function of pH and oxygen concentration. I want to say that oxygen can act as a vasodilator in some cases and a vasoconstrictor in different cases, depending on where you are. So for example, if there's a lot of oxygen in your lungs, in the blood in your lungs, it's telling your body that you're doing a good job perfusing, and you can move the blood along a little faster. If there's a lot of oxygen in the systemic circulation, it might be a sign that the cells aren't having a chance to do that diffusion, or diffusion isn't having a chance to occur yet. So you might want to dilate your vessels a little bit to allow that time. Don't quote me on that without looking it up, though, because it has been a minute since I've looked at this. As far as myogenic regulation, and this is a function of tension, which is radius times pressure. It's also cool to know that your muscles contract while walking, in the sense of squeezing your veins at least, and this pushes blood above the valve so it can't drop back down. And then if we're looking at systemic ways of controlling resistance, this is more related to neural and hormonal signals. So we're thinking things like sympathetic and parasympathetic stimulation, and then release of factors like epinephrine. It's also good to know that nitric oxide is a vasodilator in the lungs. If we look more specifically at capillaries, we can take a look at Starling's law of filtration, which essentially says that flux is a function, or it's directly proportional by some permeability constant K, and this permeability constant increases with histamines and swelling. But flux is a function of this K times the hydrostatic gradient minus the osmotic gradient. And think of the hydrostatic gradient as what's driving blood out of the vessel, out of the capillary. The osmotic gradient, what's pulling water, in this case at least, into the vessels. So to calculate the hydrostatic gradient, you'd be taking the pressure at the capillary minus the interstitial pressure. So the pressure at the capillary is kind of pressure forcing it out, and then interstitial forcing the liquid in. Capillary pressure increases a lot with pulmonary edema, which is, again, that fluid buildup in the lungs. If we're looking at osmotic gradients, this is a function of the oncotic pressure into the capillary minus out of the capillary. and flux is measured in millimeters of mercury. Also cool to know that swelling is also caused by lymphedema and that you absorb fluid from the interstitium at the midpoint during hemorrhage. If we take a look now at compliance, so this is essentially looking at the change in volume over the change in pressure. Another way of thinking of that is how much fluid or how much increased volume is it going to take for the pressure to change. Veins have very high compliance, especially related to arteries, so if you add a bunch of blood to a vein, it's not really going to change the pressure, which is very helpful for our bodies. But if compliance is low, like in the arteries, the same pressure correlates to lower volume, and you can see that in this graph over here, where at the same pressure, veins can hold a lot more volume, and a small change in venous pressure would be a large change in venous volume. So if your venous pressure changes a little bit, it's basically saying that it can take in a lot more fluid now. During pregnancy, you have increased pressure in your vessels, and this could lead to varicose veins. This is when valves fail, and we were looking a little bit at how during exercise, blood gets pushed above those valves so it can't drop back down. If blood is falling back down because the veins are failing, you can actually see them, and this is that tangled varicose vein image. Varicose veins are less compliant, they're essentially failed, and this can also happen with age. Finally, this took me a while to actually identify when I was taking the class. I struggled with this concept a little bit, but total peripheral resistance increases with sympathetic stimulation, which you'll see when Dr. Haas pulls out her cheat sheet diagram that shows the different relationships in the cardiovascular system, but TPR decreases with exercise. This is a special case. So thank you for watching. I hope this was helpful.